So we're ready to begin with Chapter 3, which is all about cells, the very basic unit of a living organism, the smallest portion of a living organism that we can think of as alive. Cells were first discovered in the late 1600s. They were discovered by a man who was working with a very primitive microscope, and remarkably, he was able to see a fair bit of stuff in the droplets of water that he was looking in. It wasn't until the mid-1800s, though, 175 years or so later, that the cell theory was formally proposed. And the cell theory, the idea that all living organisms are built of one or more cells, and it was by then generally accepted among scientists, although so there wasn't a whole lot of disagreement about most of the tenets of the cell theory when it was proposed and formally laid out. So the four tenets of the cell theory are, first, that all living organisms are built of one or more cells. So there are many living organisms that are unicellular, single-celled. Humans belong to the group of organisms, though, that are multicellular. And so our bodies are composed of billions of cells. We also recognize that cells are the basic unit of all life. So if all the cells in our body are in homeostasis, then the entire body should be in homeostasis as well, because we are built of cells, and as long as they're all functioning well, then all should be good with the body. We also all agree, as scientists, that cells can only arise from other cells. Now, I want to separate this from the thoughts or the theory of evolution by adding on the little caveat, under the current conditions, under current environmental conditions, we don't see cells self-assembling. At the point when life evolved, back 4.5 billion years ago, the conditions of the Earth were very, very different than what we experience now. There was no oxygen in the atmosphere, for one thing. And so the opportunity was there for cells to actually self-assemble. But under current conditions, we don't see that kind of production of new cells. We only see the production of new cells as other cells are reproducing, are dividing into two. And then last of all, the fourth tenet is that as a part of the processes of living, all cells must maintain homeostasis. I'm going to take a moment to do a little quick aside with this term theory. When scientists talk about a theory, they're not talking about that the same way that a layperson might be mentioning the word theory. When we say something like, "Oh, I've got a little theory about that," what we're actually, to a scientist, what a scientist would say under those conditions would be, "I have a hypothesis about that." A hypothesis is an educated guess about how something works, how some natural process works. Hypotheses only get elevated to becoming a theory, being labeled as a theory in the scientific world, when they have been tested again, retested, and they explain a very large general idea. So when scientists talk about the cell theory or the theory of evolution, these are not hypotheses, educated guesses. These are well-supported, well-thought-through statements about how we understand the world to work right now at this point. So it's almost like a law in the physical sciences. So we talk about the law of gravity, we talk about the cell theory. They are at the same level of assuredness, or we feel confident about the cell theory or about the theory of evolution in the same way that we feel confident about the law of gravity. So in the human body, there is something on the order of 75 trillion cells, which is a huge number. That is a heck of a lot of zeros that we find in the human body. What's really crazy is that we can multiply that number by 10 to think about all the single-celled organisms that are living on the human body. So we are a huge assembly, not just of our own cells, but of the community that lives on and in the cavities of the human body. And we can take these cells, if we're going to try to chunk them into different buckets based on the kind of work that they do, the characteristic, the physical characteristics of those cells, we would find that we would have to have about 260 different buckets to organize the cells into. And they all have very similar construction. Each one of these buckets, all the cells that were in those buckets, would have similar construction and function. And there is a huge variability in the size of the many different cell types. If you look in your book on page 84, you can see some of this size variability. So these are all cells that are drawn on the same scale. And we measure cell size in a unit called a micrometer or a micrometer. So 1 micrometer, and micro, the Greek letter that we use for micro is this silly looking cursive M. So that's a micro, that mu letter is that's the Greek letter mu. It's what we use for micro. A micrometer is 1/1,000,000th of a meter. So I'm not going to ask you to do any conversion between meters and micrometers, so don't expect that. I just want to give you an idea. If you think about a meter stick, it's slightly longer than a yardstick. If we take that meter stick and divide it into 1,000,000 lengths, 1,000,000 little sections, each one of those sections is a micrometer or a micrometer. Okay. So when you look then at the image on page 84, you can see huge differences. A red blood cell might be 7.5 of these micrometers, while a white blood cell could be 12 micrometers in diameter. While the egg, the gamete produced by a female human, is huge in comparison, 140 micrometers, where a muscle cell, a smooth muscle cell, this would be a muscle cell that we would find in the intestine, for instance, or the stomach. It might be 200 micrometers. Nerve cells are super long. Notice that they don't have a scale on that one. We now have a completely different scale. There can be nerve cells that have their cell body. That's the weird branchy part to the left with the nucleus inside of it. That's the cell body. That may be located in or near the spinal cord. And the long extension, called an axon, that heads out to the right, it may branch and run all the way down the leg and wind up with the little nubby tips of them hanging out somewhere in the big toe. So cells can be very, very large and lots of different shapes. So they're showing you that very sort of irregular shape associated with a nerve cell. Some cells, like the epithelial cells that line the gut cavity, may be in these flat single-layered sheets. And then, proteins, the muscle cells that were shown here, that image there in C is a cardiac muscle cell. They may have these crazy branching structures. So lots and lots of different sizes and different shapes of the cells that are found in the body. Great variability. But all of the cells in your body arose from the same single cell, an egg that had been fertilized by a sperm. So that means, so after that moment of fertilization, we now have a new living cell that has a full complement of chromosomes. And that cell divided and divided again. And those cells, so one cell divides into two cells divide into four cells divide into 8 into 16 to 32 to 64 to 128 to 256 to 500, and you get the point. It just keeps going and going. So huge numbers of cells in your body to get to that 75 trillion number, but all of them arose from the same single cell, a fertilized egg. And so that means each one carries all the same DNA, the genetic information. So DNA stands for deoxyribonucleic acid. That, if you want to know more about them, they are in Chapter 2, or we can talk in office hours, this DNA stuff. But each cell has exactly the same copy of DNA, unless there was a small mistake made. And usually those mistakes are very minor. And that DNA provides the genetic information with the instructions for all the things that the cell may need to do over the course of its life. And for all of the 260 different cell types, different instructions may be accessed. So if we think about the DNA as sort of the blueprint, some cells will go to page 3 of the blueprint and do the work that's there on page 3, or the instruction manual would be another way to think about it. While other cells might go out there to page 760, while other cells are on page 1003, while, you get the point. So all the instructions are there in every cell, but not every cell will access all of those instructions. So as cells divide and mature and divide again, there are very specific biochemical cues that tell the cell which part of the DNA to access. And so that results in the cell becoming a very distinct and specific cell type. So it's the differentiated cells. So when I say differentiated, that means cells that have gone down a developmental pathway and have access to only a small subset of all the instructions in their DNA. They can't access the other portions that are telling other cells what to do. They can only access the stuff that relates to their cell type. And their offspring cells are also going to be differentiated in the same way. Or they might even become more specific. So we'll see as we talk about, say, blood cells and discuss the development and differentiation of red blood cells and white blood cells. We'll see that there are steps after which, once that division has happened, the cell can't go backward and de-differentiate. So differentiation of cells is the access of a very specific set of instructions in the DNA and the inability to access other instructions. So we have been able to do some really cool stuff. Let's say if we think about the cells that are there at the base of the skin that are constantly dividing, replenishing the cells that are lost at the skin surface, with some very sophisticated biochemistry, scientists have been able to cause those stem cells there in the skin to de-differentiate, and then they can give them the instructions to become their offspring cells, to become a different cell type. But that is under lab conditions. That's not under natural conditions. Under natural conditions, cells do not de-differentiate. They don't go backwards. They only go forward. So on page 85, there's a picture of a composite cell. So with 260 cell types, it probably makes sense to recognize that there are no typical cells found in the body. Every cell type is very distinct from all the other cell types. But in order to get a sense of what all might be found in any one cell, we use this hypothetical composite cell to look at all the features. And so hopefully by now you've got these terms under your belt. You've been practicing and are ready for the drawing exam to occur. So let's start looking at the major parts of a cell. And so the three major parts that we can think about include the cell membrane. That's this layer and all this stuff here. So we've sliced the cell in half. We're seeing just, except for the nucleus, we've only sliced a portion of the nucleus in half. And the outer boundary of the cell is referred to as the cell membrane and has some very special qualities we'll talk about in just a second. And you can even get a close-up here of what that cell membrane looks like. The interior of the cell is referred to as cytoplasm. And that is the watery gel, the cytosol. Think cyte, by the way, here. When you see that prefix, what does that mean? Hopefully you just said cell. CYT means cell. If we talk about the cytosol, that's the solution, the fluid that is inside the cell. So when we talk about cytoplasm, we're talking about the gel, the liquidy parts, and the organelles. Organelles, that's like the little organs. These are the machinery, the little structures inside the cell that do the work. So everything inside the cytoplasm, inside the cell membrane, is cytoplasm, with one small exception, and we characterize the nucleus or classify it outside of cytoplasm. It's another location inside the cell. There's the cytoplasm with the cytosol and the organelles, and then there is the nucleus, and that's the blue part here, that contains the cell's DNA. And it's going to be really tempting to call this small round structure inside the nucleus the place where the DNA is, but that's not the truth. These little squiggly lines here that fill up the nucleus, those are the strands of DNA. And external to the cell is bathed in watery extracellular fluid. Extra means beyond, cellular, obviously. So extracellular means anything that's beyond the cell. And what we find outside of a cell is fluid. If there's no fluid outside the cell, the cell's going to be in really bad shape. It will be dead, or if not dead already, it will be almost dead if there were no fluid around that cell. So the cell membrane has some very, very specific characteristics. It's flexible and elastic, and it serves as a barrier between the internal or intracellular environment and the external or extracellular environment. We talk about the cell membrane as being selectively permeable. If something is permeable, that means it allows things through. If it is 100% permeable, that means anything can pass through it. So if you open the door, you have now made the room permeable to anything that will pass through the door. But for something to be selectively permeable, that means that some things are allowed in, other things are kept out. So it would be like opening the door but draping a net over the door. And that way people could get in and out around the net, but the small flying insects, the mosquitoes, the wasps, they would be excluded from coming into the room. So a selectively permeable characteristic, this characteristic of selective permeability, means that the cell membrane gets to regulate what comes into the cell and what leaves the cell, to a large part. There's some things that can get through without they are invisible to the cell membrane. The cell membrane is also very sensitive to the environment. It notices what's going on in that extracellular environment because it's got receptors on the membrane surface. And these receptors are built in a way that they will recognize specific molecules. And so if a specific molecule lands in one of these receptors, then it can trigger some new function inside the cell. We would talk about that as signal transduction. And I believe I've introduced that term to you already. We're going to look at that a lot more when we get to the chapters on the nervous system and the endocrine system. So let's look closely now at the cell membrane structure. It's composed primarily of 3 molecules. The greatest portion of it are lipids. And there's a specific type of lipid. It's called a phospholipid. A phospholipid looks a lot like a dietary fat, but we've stripped off a fatty acid from that dietary fat. So dietary fats look like this. There's a carbon backbone with an oxygen, and then attached to it is this long string of carbons called a fatty acid. So that would be, in general, what a dietary fat looks like. But if we cut off the fatty acid, so we get rid of this fatty acid, and instead attach a phosphate group then we create a phospholipid. So PO4, that's how I'm designating a phosphate group. If you have questions about this, you might want to look back at Chapter 2 or meet with me during office hours. So this dietary fat with the phosphate group in place of one of the fatty acids kind of reorganizes itself so that it has a head region. This is where the phospholipid is. So that would be this part up here and some of this here, kind of all clumped together. And then hanging off of that head are two tails that are just composed of carbons. So that's a phospholipid. And these phospholipids are going to be arranged in what we talk about as a bilayer. We'll talk about that in just a second. We also see kind of floating and able to move around in this ocean of phospholipid, these floating islands that are made up of primarily proteins, but sometimes we can have, decorating those proteins, carbohydrates. And sometimes we see, decorating those proteins, other kinds of lipids. So lipids, proteins, carbohydrates, all Chapter 2 stuff that we skipped over. So if you're not comfortable with those concepts or those ideas, be sure to check out Chapter 2. So when we look really close at a phospholipid, we notice that the head, so that's the phospho part, here's the phosphate group, the PO4 part, it is built in a way that it will play around with water. It is hydrophilic. Phil means liking, hydro means water. So that's the water-liking portion of a phospholipid. It interacts with water, and it will always be oriented to the area of the environment that has a watery component to it. Attached to the hydrophilic head are two long tails that are nonpolar or hydrophobic. Phobic means fearing, so these tails are water fearing, and they are repelled by water. They do not interact with water. They're the oily part of the cell membrane. And as you know, the oil and water, they don't mix. Water is polar, just like the head is polar. So polar things will play and like to play with other polar things, while the tails are what we call non-polar. Again, this is Chapter 2 material, or hydrophobic, because non-polar things do not like to play with polar things. So when you throw phospholipids into a watery bath, they will automatically organize themselves because of this hydrophilic and hydrophobic nature of a phospholipid. And the way that they organize themselves is with the tails orienting toward each other and the heads orienting toward the watery environment. So here we have the extracellular fluid, the ECF. Here we have the cytosol, both watery environments. The hydrophilic heads are oriented and interacting with the water that's in the cytosol, while the hydrophobic tails are going to orient toward each other, interact with each other, creating this region here that has a very oily characteristic to it. So anything that can dissolve in lipids will pass through a cell membrane very easily. But things that will dissolve in water and not lipids, they are stuck. They cannot move from one side of the cell membrane to the other unless we do something else to it. So lipid-soluble molecules pass, water-soluble molecules don't pass. That creates, or that is, the selectively permeable characteristic of the cell membrane. And it's all because the tails are oily and let oily things come through while the heads are not. And now, Okay. I was about to say something and decided not to. All right, so hopefully that makes sense to you now. And the book has more great pictures, just like the one that I have been showing. Well, yeah, that's right. The new book does not have a lot of information there. And I'm going slightly out of order with the book, which maybe let me just double check when I say that. No, we're good. Yeah. All right. So we also find some other lipids in this layer, and those lipids are referred to as cholesterol. You've heard of that before. This is a really important component of a cell membrane because these cholesterol molecules, and here they are, the cholesterol molecules are here and here, they help to provide rigidity and stabilize the lipid molecules there by interacting with them. And then there could be lipids called glycolipids. These are lipids with attached carbohydrates here that are found in the ocean of the cell membrane, the lipid portions of the cell membrane. So what else is found in the cell membrane? Well, now we've got to look at the proteins that are found there. And there are several different kinds of proteins that we can talk about. There are integral proteins. These are proteins that are found embedded in the membrane. I should not, they don't. Let me try that again. They are embedded in the membrane. So here is an integral protein. Here is another integral protein. They both have a face or a portion of their protein that is in contact with the oily inner membrane or inner layer of the cell membrane. Peripheral proteins are found just at the surface, and they could be either inside or outside, so they don't extend down into the membrane itself. And a transmembrane protein is an integral protein that it has a surface both outside and inside the cell. So they project out or into the cytoplasm, and they project out into the extracellular fluids. So these proteins have different functions. They could be receptor proteins. These are the transmembrane proteins that combine specific molecules outside the cell and then create a change inside. So if a molecule comes and binds here at this receptor site, that will change the structure of the integral receptor protein, that transmembrane protein, and causes something new to happen inside the cell, at the surface that's inside the cell. Or they could be channel proteins. These are transmembrane proteins that allow things that would not normally pass through the membrane. Dadgummit, that should be hydrophilic. So water-loving materials that cannot get through the membrane. So it allows hydrophilic materials a pathway into the cell. And it could be because they're like an open pore through the cell membrane formed by that channel protein. Or it could be a closed channel that can open up under specific circumstances. So again, these channel proteins are transmembrane proteins that allow hydrophilic materials to cross through that oily inner layer of the cell membrane, because they make a little hole or a channel or that allows the material, that hydrophilic material, to just slide right on through the hydrophobic interior of the membrane. Some other functions that these things can have: they might be cell adhesion molecules, known as CAMs, C-A-Ms. These are peripheral proteins that will attach between themselves. So they're found on two different cell surfaces, and so they will attach to each other and bind the two different cells close to each other, these cell adhesion molecules. And they may be glycoproteins. These are peripheral proteins that have attached carbohydrates, and they could be out there sending a message saying, hey, I belong in this body. Don't mess with me unless something else has gone wrong. Or they could be something that the body doesn't recognize, in which case they tell the cell, a cell that might be checking, that, hey, this cell doesn't belong in the body. So glycoproteins are used as identifier proteins. They are generally peripheral proteins, and it's a carbohydrate that helps to allow any cell that's checking to know whether that's a self cell or a non-self cell. All right, so that's it for this lecture. So just to review, we talked about the cell theory and the 4 tenets of the cell theory, and then we talked about the materials that are used to make up the cell membrane and the lipids, proteins, and carbohydrates, and the very specific characteristics of phospholipids and why that creates a selectively permeable membrane that helps to control what is inside and what is outside of a cell. All right, any questions? Talk to me in class, or find me in office hours, or send me a message, email.